Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 3, Tools of Recombinant DNA Technology. Tools of Recombinant DNA Technology. Now we know from the foregoing discussion, the genetic engineering or recombinant DNA technology can be there accomplished only we have key tools. So how to the recombinant DNA technology? So before when we are using as in the total to make as in one recombinant DNA, we are required as a some tools. Am I right? See what type of tools we require. Let's be down. See the first one is key tools. First one is restriction enzyme. Second one is polymerase enzyme. Third one is ligases. Fourth one is vectors. And fifth one is what? Host organism. So this 5 tools is required for as into development of our DNA. Okay. Next, let us try to understand some these in detail. Okay, let's discuss in one by one detail. Okay, now the first one is what? The first one is restriction enzymes. First one is what? Restriction enzymes. See, in this academic year in 1963, the two enzymes they are responsible for the restriction of the growth of Bacteriophage Escherichia coli was isolated. In 1963, the two enzymes is responsible. The restriction growth of Bacteriophage E. coli was isolated. Escherichia coli is isolated. One of these added the methyl groups or to DNA. So the first when we are taken as the DNA to get add with as what? In a methyl group. And later it is called as a restriction endonuclease. So then while the DNA we are cut and lateral we call as what in the restriction endonuclease. So when you uptake this DNA, when you are add with as a methyl group of the DNA, we cut the DNA into as in specific locations, that is a restriction endonuclease. Next, after this first restriction endonuclease, that is HIND2. The first very important restriction endonuclease is what? HIND2, whom they are functioning the depend on the specific DNA nucleotide. The sequence was isolated and the characteristic 5 years later. So after the first the restriction endonuclease HIND2, then the DNA nucleotides they are isolated the characteristic after few years 5 years later. Next, it was found that HIND2 always cut the DNA molecule in a particular point of the in a recognizing specific sequence of 6 base pairs. That specific base pair sequence is known as their recognition sequence for HIND2. For HIND2. So where the total DNA we are completely they are cutting. So where we are cutting the specific location, that the specific location when we are cutting, so that specific region is called as what? That is in a recognition sequence for HIND2. Okay. Next, beside the HIND2, today we are no more than 900 restriction enzymes is present. More than here 900 restriction enzymes that we have isolated from over in a 230 strains. 230 strain of bacteria, each one recognized different recognition sequence. So when we are taking as in that completely 230 strains, each bacteria has in that different recognition site is present. Okay. Next, after here the conversion for naming these enzymes, the first letter of the name comes from the genus and the second letter, 2 letters comes the species of the prokaryotic cell from which they are isolated. The first one is what? The first name comes as in a that genus. The second letter, 2 letters, that is what? One is in a species. The second one is what? That is a prokaryotic cell. Is not which they are isolated. Example, they give an EcoRI. They given as an EcoRI. It comes later the Escherichia coli RY and 13. Here EcoR1, that is R is derived from the name of their strain. Here R derived from the name of here their strain Roman number that following the names indicates as any other which as an enzyme is where isolated from any strain of bacteria. So after we easily to where from we are isolated, that is the strain of the bacteria. Next, restriction enzymes belongs to as a large class. Enzymes is called as a nuclease. So the restriction enzymes we are placed as in the large class. That class name is what? Nuclease. There are two kinds of exonuclease and endonuclease. The two classes, so the one is for as an exonuclease and another one is what? Endonuclease. The exonuclease removed nucleotides from as an end of the DNA, where the endonuclease that makes cut the specific position of the DNA. So mostly the exonuclease where we are cut the DNA into as an their end of the DNAs, and the endonucleus it cut as in a specific location within as in a DNA. So in the specific locations only means in the specific locations only the mostly the DNA will be they get cut. Okay, now that is in a exonucleus and then endonucleus. Okay, next, each restriction endonuclease functional they are inspecting of the length of DNA the sequence. The first here what is the first role? Here the DNA sequence, the first the endonuclease function is what? The inspecting. So which kind of that sequence is present? Where the DNA, what is the length of the DNA sequence? That is first function. Next, once find the specific recognition sequence, it is will bind the DNA and cut the each two strands to double helical specific points there in a sugar and phosphate backbone. So first to get identified, after to get identified in this total sequence will be they get once to get recognized, then what happened? They get start with as in their DNA cut. Okay, in the DNA cut with as in specific locations. Here in the total DNA sequence also where that mostly the sugar and phosphate groups is present, they cut their DNA. Each restriction endonuclease recognition specific palindromic nucleotide sequence in the DNA. So when we are cut the total DNA, the DNA also to get complete to get followed by as in a what? Palindromic sequence of the DNA. What is a palindromic? See here when we see as in that both backward reaction and then forward reaction both are same. Here mostly there backward reaction as well as in a forward reaction both are same, that is a part of the in a palindromic sequence. Action for restriction enzymes. So they given here clearly the enzymes cut both DNA strands at the same site. So they cut here guanine, adenine, adenine, thymine, thymine, cytosine, guanine, adenine, adenine, thymine, thymine, cytosine. Like here, EcoR1 cuts the DNA between as a guanine and adenine. The mostly we cut the DNA. See guanine and adenine sites. Guanine and adenine sites only the DNA we get cuts. Okay, na? That is next. See after they cut the see they given here clearly the sticky ends also here also sticky ends forms, here also they get sticky ends forms. Okay, so these both are sticky ends we get joined with help of what? DNA ligase enzyme we developed as in one rDNA. rDNA means recombinant DNA we are forms. Okay, now that is here. Do you know that what is the palindromic here? They are groups of the letters that form as in same words where they read the both forward and backward reaction. So both backward and forward the words will be the same. That is called as what? Completely called palindromic sequence. They given here Malayalam. They given as what? Malayalam. In front to back and back to front. If they having as the same sequence is present. The against words the palindromic where as in the same words in read both directions. Palindrome in a DNA sequence, the base pairs read the same of the two strands of in origin reading kept as in a same. Here they given following there for example following sequence here reads the same on the two strands 5 prime to as in 3 prime one direction. It is also true if read as in a 3 prime to as in a 5 direction. So one is in a 5 to 3 and second one is in what? 3 to 5 direction. So they given here clearly GAA TTC, they are GAA TTC. Okay, this is. Let's Here, the restriction enzymes cut the strand of the DNA a little way from a center of palindromic sites. So the palindromic sites only the most of the DNA will get cut. But between the both base of the opposite strands. So here most of the both strands, most of the both strands. So they given here clearly. So it is in a one DNA strand, it is also one DNA strand. So in that specific regions only we cut the DNA. So for example, they cut here, we cut as in a DNA particle here, and if we cut here as in a that in these regions. Okay, so then after when you cut the DNA, they makes like, okay, and see this level of the single stranded portion of at the end there are overhanging the stretches called as in sticky ends. So when we are cut that and the enter these regions are called as what in a sticky ends. Each of the stand these are named because they are in a hydrogen bond. So in that regions they having as what in that region has in a mostly hydrogen bonds is present. So in that sticky end regions, in that sticky end regions, they having as what completely hydrogen bonds is present. Okay, and after see there has any their complementary cut has any counterparts. Now this sticky and the stickiness here, the stickiness of the end the facilitate at their action of the enzyme is DNA ligase. So when this cut the DNA here, how do we get facilitates with the help of the enzyme is what DNA ligase. DNA ligase. So we are using a DNA ligase. So here we completely to get add in that DNA piece into as a vector region. Next, the restriction endonuclease are used as in genetic engineering to form as a recombinant molecule of DNA. So finally, we're getting as what the new DNA is coming. That is a rDNA, recombinant DNA, which is composed of the DNA from as any their different source of genome. So when we are getting this DNA is present, the DNA is called as what? Recombinant DNA. That is called as what? Recombinant DNA. Let us when you cut the same restriction enzyme. Result to DNA fragments have the same kind of the sticky ends designed joined which together end to end using as a DNA ligase enzyme. So here foreign DNA as well as in a vector DNA we cut and they get add with as an A here with the help of the DNA ligase enzyme. So finally we are getting as what rDNA is comes. So rDNA is converted as a transformation cells and the cell means the E. coli cell it is a completely divided into as in two cells. They are completely host the cloning host cells. Okay, now it is in a diagrammatic representation as an A or DNA recombinant DNA technology. Okay, let's after you have in a next you have the realized that normally the unless one cut the vector and the source of the DNA while the same the restriction enzymes recombinant their molecule cannot be there created. So without this or we can't to create as a new DNA fragments. Okay, this is so remaining we discuss in next class. Thank you for watching. Please share and subscribe. Thank you.